Close your eyes for a moment and picture a forest. Perhaps you see towering trees, their leaves catching sunlight. Maybe you notice birds flitting between branches, or a deer stepping carefully through the undergrowth. But beneath your feet, invisible to the casual observer, stretches a kingdom more vast and interconnected than anything you can see above ground. Tonight, we're going to explore that kingdom. Welcome to Bedtime Biology, where science meets serenity. Each night, we explore the quiet wonders of the natural world, calming your mind and sparking your curiosity. Please, like, subscribe, and comment below with where you're checking in from. We're stepping into the hidden realm of fungi, organisms so fundamental to life on Earth that without them, the world as we know it would simply cease to function. They are not plants, though we once thought they were. They are not animals, though they share more in common with us than with the green things that grow towards sunlight. They occupy their own kingdom, separate and ancient, diverse beyond measure. Fungi have been here far longer than we have. They were among the first life forms to colonize land, hundreds of millions of years ago, working alongside early plants to make the terrestrial world habitable. Today, scientists estimate there are somewhere between 1.5 million to nearly 4 million species of fungi on Earth. Yet, we have identified and named only about 144,000 of them. The vast majority remain unknown, growing quietly in soil, on trees, within rocks, beneath ice, in the deepest parts of the ocean, and even in the upper atmosphere. What makes a fungus a fungus? The answer is more complex than you might think, but let's start with the basics. Fungi are eukaryotes, meaning their cells contain a nucleus and other membrane-bound structures, just like our own cells. But unlike plants, fungi cannot photosynthesize. They cannot capture sunlight and transform it into energy. Instead, they must absorb their food from their surroundings, secreting powerful enzymes that break down complex organic matter into simpler compounds that their thread-like bodies can absorb. This mode of feeding places fungi in a unique ecological role. While plants are producers, creating energy from sunlight, and animals are consumers, eating other organisms, fungi are primarily decomposers, breaking down the dead and recycling nutrients back into ecosystems. Without them, dead plant and animal matter would accumulate endlessly. Forests would choke on their own fallen leaves. The carbon cycle would grind to a halt. Life as we know it depends on these quiet decomposers working ceaselessly, unseen, beneath our feet. The invisible architecture let us begin by understanding the structure of these remarkable organisms. When you think of a fungus, you might picture a mushroom, that familiar cap and stem structure you find in grocery stores or growing in rings in your yard. But that mushroom is merely the fruiting body, the reproductive structure that the fungus produces when conditions are right. It's the equivalent of an apple on an apple tree. The real body of the fungus, the part that lives and feeds and grows, lies hidden. That hidden body is called the mycelium. Picture a network of incredibly fine threads, each one thinner than a human hair, branching and rebranching, spreading through soil, through dead wood, through whatever substrate the fungus calls home. Each individual thread is called a hypha, and collectively, these hyphae form the mycelium. The word comes from ancient Greek, meaning fungus, but it evokes something of the nature of these organisms, their web-like interconnected structure. A single fungal hypha is a marvel of biological engineering. Unlike plant cells, which have walls made of cellulose, fungal cell walls are constructed primarily of chitin, the same tough, flexible material that makes up the exoskeletons of insects and the shells of crustaceans. This gives fungal cells remarkable strength and flexibility, allowing them to penetrate solid materials, to push through soil, to invade wood, to grow in places where other organisms cannot. The hyphae grow from their tips, extending forward cell by cell, following chemical gradients toward food sources. This process is called chemotropism, the growth toward or away from chemical signals. When a hyphae detects the presence of nutrients, perhaps the scent of decaying leaves, or the chemical signature of dead wood, it grows in that direction, extending and branching, sending out more hyphae to colonize the food source more thoroughly. As the mycelium spreads, the hyphae begin to anastomose, which is to say, they fuse with one another, creating cross-connections that form a three-dimensional network throughout the substrate. This network is not random. It has structure, purpose, efficiency. The fungus is solving a problem, the problem of how to extract maximum nutrients from its environment while expending minimum energy. The solution is this web, this net, this living architecture that can span enormous areas. How enormous? In a single teaspoon of forest soil, you might find several meters of fungal hyphae. In a single gram of soil, the hyphae can stretch for more than 200 kilometers if laid end to end. The density is almost incomprehensible. And when you scale up from a teaspoon to an entire forest, the numbers become astronomical. The mycelium forms a living mat beneath the forest floor, permeating everything, touching everything, connecting everything. But fungi are not uniform. The kingdom is incredibly diverse, split into several major divisions based on how they reproduce and how their hyphae are structured. The ascomycetes, or sac fungi, include species like baker's yeast, the molds that make penicillin, the fungi that give us morels and truffles. The basidiomycetes encompass most of the familiar mushrooms, the bracket fungi that grow on trees, the puffballs that release clouds of spores when stepped upon. The zygomycetes include the bread molds, the fuzzy growth you find on forgotten fruit. Each division has its own characteristics, its own evolutionary history, its own ecological roles. Within their cells, fungi store not glycogen, which is what we use, but they also use glycogen like animals do, another way in which they differ from plants. They accumulate lipids as energy reserves. They produce melanin, the same pigment that colors human skin, which protects them from ultraviolet radiation and helps them survive in harsh environments. Some fungi can even produce compounds that act as antifreeze, allowing them to remain active in sub-zero temperatures. The relationship between fungi and their environment is intimate and complex. Fungi don't simply sit on top of their food source and absorb nutrients. They actively transform their surroundings, secreting enzymes into the substrate that break down cellulose, lignin, proteins, chitin, and countless other compounds. These enzymes are excreted outside the cell, performing extracellular digestion. Then, the products of that digestion are absorbed through the hyphal walls. This ability to produce such a diverse array of enzymes is what makes fungi such effective decomposers. Lignin, the tough, complex polymer that gives wood its strength and rigidity, is extremely difficult to break down. Few organisms can do it. But certain fungi, the white rot fungi and brown rot fungi, have evolved enzymatic systems specifically for degrading lignin, allowing them to decompose wood and return its nutrients to the ecosystem. The speed at which fungi can colonize and break down organic matter varies enormously depending on the species, the substrate, and environmental conditions. Some fungi work slowly, taking years or decades to decompose a fallen log. Others work with startling speed, sending out hyphae that can grow several centimeters per day under ideal conditions, colonizing fresh substrates rapidly before competitors arrive. And competition is fierce in the fungal world. Multiple species of fungi often inhabit the same substrate simultaneously, each trying to claim resources for itself. They engage in chemical warfare, producing antibiotics and other toxic compounds to inhibit or kill competing fungi and bacteria. This is not aggression for its own sake, but simple evolutionary necessity. The fungus that can most effectively claim and defend its food source is the fungus that survives to reproduce. This brings us to reproduction, and here again, fungi display remarkable diversity. Many fungi can reproduce both sexually and asexually. Asexual reproduction is straightforward. The mycelium simply fragments or produces spores mitotically, creating genetic clones of itself. This is efficient and allows rapid colonization of suitable habitats. Sexual reproduction is more complex, involving the fusion of specialized cells from two different individuals, followed by nuclear fusion and meiosis to produce genetically diverse spores. The fruiting bodies we recognize as mushrooms are sexual reproductive structures. They develop when two compatible mycelia encounter each other and fuse, and when environmental conditions, particularly moisture and temperature, are favorable. The mushroom emerges from the substrate, expands, and releases spores by the millions or billions. These spores are carried by wind, water, or animals to new locations where, if they land in a suitable habitat, they germinate and begin growing new mycelia. Spore production is prodigious. A single giant puffball can produce several trillion spores. A large mushroom might release billions. The vast majority of these spores will die, landing in unsuitable locations or being eaten by other organisms. But enough survive to ensure the continuation of the species. This is life's ancient strategy. Produce in vast quantities, and let chance and selection determine which individuals will survive to reproduce in turn. In the darkness, in the stillness, the mycelium grows. Invisible to our eyes, imperceptible in its movement, it extends through the soil, following the scent of nutrients, building its network, preparing for the moment when conditions align and it can send up its fruiting bodies into the world above. Patient, persistent, endlessly adaptable. This is the fundamental architecture of the fungal kingdom, the underground internet. Beneath the forest floor, something remarkable is happening. Something that scientists only began to understand in the late 20th century, though indigenous peoples and careful observers of nature had sensed it for much longer. The trees in a forest are not isolated individuals, each competing against the others for resources and survival. They are connected, linked by an underground network of fungal hyphae that allows them to communicate, to share nutrients, to support one another. This network has been called the Wood Wide Web, a clever play on words that captures something essential about its function. Like the internet that connects computers around the world, allowing them to exchange information, the mycorrhizal network connects the plants in an ecosystem, facilitating the transfer of carbon, nitrogen, phosphorus, water, and even warning signals between individuals. The story begins with mycorrhiza, a word that means fungus root in Greek. Mycorrhizae are symbiotic associations between fungi and plant roots, relationships in which both partners benefit. The plant provides the fungus with sugars produced through photosynthesis, while the fungus provides the plant with water and mineral nutrients absorbed from the soil. These relationships are ancient, dating back hundreds of millions of years to when plants first colonized land. It's estimated that roughly 90% of all land plants form mycorrhizal associations, from tiny mosses to towering sequoias, from alpine flowers to tropical palms. Plants have evolved in partnership with fungi. In many cases, plants literally cannot survive without their fungal partners. Seedlings growing in soil sterilized of fungi often struggle to thrive, even when given adequate water and nutrients, because they lack the enhanced absorption capacity that the fungal network provides. There are several types of mycorrhizae, but the two most important are arbuscular mycorrhizae and ectomycorrhizae. Arbuscular mycorrhizal fungi belong to an ancient lineage called glomeromycota. They penetrate the cells of plant roots, forming branching structures called arbuscules that dramatically increase the surface area for nutrient exchange. These fungi are particularly important in grasslands and agricultural systems, forming associations with most crop plants, including wheat, corn, and soybeans. Ectomycorrhizal fungi, by contrast, do not penetrate root cells, but instead form a sheath around the outside of fine roots and extend hyphae into the spaces between root cells. These associations are particularly common in temperate and boreal forests, forming partnerships with trees like pines, firs, oaks, birches, and beeches. Many of the familiar forest mushrooms, chanterelles, boletes, amanitas, are the fruiting bodies of ectomycorrhizal fungi. But mycorrhizae do more than simply help individual plants absorb nutrients. A single mycorrhizal fungus can connect with multiple plants simultaneously. And because many fungi species coexist in the same soil, each plant is typically connected to multiple fungal partners. The result is a vast, complex network that links most or all of the plants in a community. This discovery was made in the late 1990s by forest ecologist Suzanne Simard and her colleagues at the University of British Columbia. Simard grew up in the forests of British Columbia, where her family had worked as loggers for generations. When she became a scientist, she began investigating questions that arose from her observations of how forests actually behaved, as opposed to how forestry practices assumed they should behave. At the time, conventional forestry wisdom held that conifer seedlings, the commercially valuable trees like Douglas fir, competed with deciduous trees like paper birch for resources. The solution, according to this thinking, was to eliminate the birch using herbicides, giving the conifers more room to grow. But Simard noticed that forests where birch had been removed did not necessarily perform better. Something else was going on. To investigate, she designed an elegant experiment. She grew Douglas fir and paper birch seedlings in close proximity and supplied one species with carbon dioxide containing radioactive carbon-14, while supplying the other with carbon dioxide containing stable carbon-13. Then she waited to see if the carbon would move between the trees. It did. The carbon moved from one species to the other through underground fungal connections. Moreover, the direction of carbon flow changed depending on conditions. In summer, when the birch was fully leafed out and photosynthesizing vigorously, carbon moved from birch to fir. In autumn, when the birch was preparing for winter dormancy, carbon moved the other direction. The trees were actively sharing resources through the mycorrhizal network, supporting each other through different seasons. Subsequent research revealed even more sophisticated behaviors. Older, larger trees, which Simard called mother trees, serve as hubs in the network, connecting to dozens or hundreds of other trees and seedlings. These hub trees can recognize their own offspring through chemical signatures and preferentially send carbon and nutrients to their kin. When a tree is dying, whether from disease, insect attack, or old age, it often releases stored nutrients back into the mycorrhizal network, effectively bequeathing its resources to the next generation. The network also transmits warning signals. When a tree is attacked by insects or infected by disease, it can send chemical signals through the mycorrhizal network to neighboring trees, prompting them to increase production of defensive compounds before the threat arrives. This early warning system gives the community an advantage, allowing trees to mount defenses preemptively rather than reacting after they've been attacked. The implications of this research are profound, both scientifically and philosophically. It challenges our fundamental assumptions about plants as passive, competitive organisms. Instead, we see evidence of cooperation, communication, even something that might be described as intelligence, though not consciousness in any sense we would recognize. The forest emerges not as a collection of individuals, but as a superorganism, a community united by underground connections. But what's in it for the fungi? Why would they facilitate these transfers of nutrients between plants? The answer lies in the fungus's own evolutionary interests. The mycelium requires a steady supply of carbon in the form of sugars, which it obtains from its plant partners. By keeping multiple plants healthy and productive, the fungus ensures its own food supply remains stable and abundant. If all the plants in the network are thriving, they can collectively provide more sugars than any single plant could alone. Moreover, a diverse network is more resilient than a simple one. If the fungus partners with only a single plant species, and that species is wiped out by disease or environmental stress, the fungus loses its food source. But if the fungus maintains connections with multiple species, occupying different niches and responding to different stresses in different ways, the fungus's survival is more secure. Biodiversity benefits everyone. The mycorrhizal network is not always benevolent. Parasitic plants can tap into the network, stealing carbon from other plants without contributing photosynthesis of their own. Some fungi cheat, taking carbon from plants without providing nutrients in return. Like any complex system, the Wood Wide Web contains both cooperation and conflict, mutualism and exploitation. It reflects the full complexity of ecological relationships. The physical extent of these networks can be enormous. In a forest, every step you take covers ground permeated by fungal hyphae connecting hundreds of plants. The finest roots of trees, the root tips where nutrient absorption occurs, are so intimately associated with fungal hyphaes that it's often difficult to tell where root ends and fungus begins. They grow together, function together, have co-evolved together for so long that they are, in many ways, a single organism. Research has shown that the mycelial network is not merely passive infrastructure, but actively manages nutrient distribution. The fungus appears to allocate resources based on complex signals, sending nutrients to plants that need them most, responding to changes in supply and demand across the network. Some researchers describe this as a kind of market system, with the fungus acting as broker, while others see it as a more centralized distribution network. The true mechanisms remain an active area of research. In laboratory experiments, researchers have measured the electrical signals that travel through fungal mycelia, pulses of activity that resemble, in a very crude way, the electrical impulses in animal nervous systems. These signals appear to coordinate fungal behavior, communicating information about food sources, damage to the mycelium, and other environmental conditions. Whether this constitutes something we might call communication or cognition is a matter of ongoing debate, but it's clear that fungi are processing information about their environment and responding in sophisticated ways. The mycorrhizal network even influences soil structure itself. The hyphae bind soil particles together, creating aggregates that improve soil porosity, water retention, and resistance to erosion. The fungus secretes compounds, proteins called glomalin, that act as biological glue, holding the soil matrix together. Over time, this fungal engineering creates soil structure that benefits the entire ecosystem. When we walk through a forest, we see trees, ferns, flowers, the visible life. But beneath our feet, filling the spaces between soil particles, infiltrating every root system, connecting plant to plant and plant to fungus in an elaborate web of mutual dependence, the mycelium grows. Silent, invisible, essential. This is the forest's foundation, its underground architecture, the wood wide web that makes the forest possible. Every tree you see is part of this network, connected to every other tree, sharing and receiving, giving and taking, bound together by threads of living fungal tissue that weave through the darkness of the soil. Masters of decomposition, imagine, for a moment, a world without decomposition. Every leaf that ever fell would still lie on the ground where it landed. Every tree that ever died would remain standing or fallen, but never recycled. The bodies of every animal that ever lived would pile up endlessly. The Earth's surface would be covered in dead organic matter, dozens of meters deep, perhaps deeper. No new growth would be possible because all the nutrients would be locked away in dead tissue, unavailable to living organisms. This nightmarish scenario doesn't happen because of decomposers, and among decomposers, fungi are supreme. While bacteria also play important roles in decomposition, fungi are uniquely equipped to break down the most recalcitrant, difficult to decompose materials, particularly the lignin and cellulose that make up plant cell walls and give wood its strength. Let's understand what makes decomposition so challenging and why fungi are so good at it. Plant matter, especially woody plant matter, is composed primarily of three polymers: cellulose, hemicellulose, and lignin. Cellulose is a long chain of glucose molecules linked together in a particular configuration. It's the most abundant organic polymer on Earth, and many organisms, from bacteria to fungi to certain animals, have evolved enzymes called cellulases that can break it down. But lignin is different. Lignin is a complex, irregular polymer made up of aromatic rings linked together in seemingly random ways. It's what makes wood hard and resistant to decay. It's what allows trees to grow tall and strong. And it's extraordinarily difficult to break down. Very few organisms possess the enzymatic machinery necessary to degrade lignin effectively, and most of those organisms are fungi. The fungi that specialize in decomposing wood are divided into two major categories based on the color of the residue they leave behind. White rot fungi decompose both cellulose and lignin, leaving behind pale, fibrous material that crumbles easily. Brown rot fungi preferentially attack cellulose and hemicellulose, while leaving lignin relatively intact, resulting in brown, cubic chunks of degraded wood that break apart along straight lines. The enzymatic systems these fungi use are marvels of biochemical engineering. White rot fungi produce powerful oxidative enzymes, including lignin peroxidases and manganese peroxidases, that generate highly reactive free radicals. These free radicals attack the lignin polymer at random points, breaking the bonds that hold it together. It's a brute force approach, using chemical aggression to tear apart a molecule that's resistant to more subtle enzymatic attack. Brown rot fungi use a different strategy, producing enzymes and compounds that generate hydroxyl radicals and other reactive oxygen species. These attack cellulose from the outside, breaking it down into simpler sugars while leaving the lignin matrix largely intact. The result is different, but the outcome is the same. Complex plant material is transformed into simpler compounds that can be absorbed by the fungus and eventually returned to the soil. But decomposition is not only about breaking down wood. Fungi decompose leaves, needles, roots, fruit, animal remains, and myriad other organic materials. Each type of substrate requires slightly different enzymatic tools, and fungi have evolved an enormous diversity of enzymes to meet these challenges. Some fungi specialize in particular substrates, becoming highly efficient at decomposing that one type of material. Others are generalists, capable of decomposing many different things, but perhaps not excelling at any one. The speed of decomposition varies enormously, depending on the fungus, the substrate, and environmental conditions. A leaf might decompose in a few months, with most of the breakdown accomplished by bacteria and fungi working together. A fallen log in a temperate forest might take a decade or more to fully decompose, with succession of different fungal species colonizing it over time, each breaking it down a bit more. In cold climates or very dry environments, decomposition slows dramatically, and organic matter can persist for decades or centuries. Temperature and moisture are critical. Fungal enzymes work within certain temperature ranges, typically performing best between 10 and 30 degrees Celsius, though some fungi have adapted to colder or warmer conditions. Moisture is essential because fungal hyphae need water to function, and the enzymatic reactions of decomposition require an aqueous environment. When conditions become too dry or too cold, decomposition effectively stops, and the organic matter enters a kind of suspended animation, waiting for conditions to improve. Decomposition is not a solitary activity. Fungi often work in consortium with bacteria, each complementing the other's activities. Bacteria are particularly good at breaking down simple sugars, amino acids, and other readily digestible compounds. Fungi excel at attacking more complex materials. Together, they can decompose organic matter far more efficiently than either could alone. The mycelium provides physical structure through which bacteria can move, while bacterial metabolism can create conditions that benefit fungal growth. Soil invertebrates also play crucial roles. Earthworms, springtails, mites, nematodes, and countless other tiny animals graze on fungi, breaking up mycelium and dispersing spores. This grazing can actually stimulate fungal growth by inducing the mycelium to produce new hyphae to replace those that were consumed. The animals also break down organic matter physically, chewing it into smaller pieces that provide more surface area for fungal and bacterial attack. The result of all this activity is soil, that dark, rich, living substance that covers much of the Earth's land surface. Soil is not simply broken down rock. It's a complex matrix of mineral particles, organic matter in various stages of decomposition, living organisms, water, air, and the biochemical products of countless biological processes. Fungi are architects of soil, building its structure through their hyphae, enriching it with the nutrients released by decomposition. Through decomposition, fungi accomplish something essential. They complete the nutrient cycle. Plants take up carbon dioxide from the atmosphere and mineral nutrients from the soil, using sunlight to combine them into organic matter. When plants die, decomposers break that organic matter back down, releasing carbon dioxide back to the atmosphere and returning mineral nutrients to the soil, where plants can use them again. Without this recycling, ecosystems would quickly exhaust their available nutrients and collapse. Consider nitrogen, an element essential for all living things, a component of amino acids, proteins, and nucleic acids. Plants can absorb nitrogen only in certain chemical forms, primarily ammonium and nitrate. When organisms die, their proteins and other nitrogen-containing compounds are locked away in complex organic molecules. Decomposers, including fungi, break down these molecules, releasing nitrogen in forms that plants can use. This mineralization of organic nitrogen is absolutely crucial for maintaining ecosystem productivity. The same is true for phosphorus, another essential nutrient that's often in short supply. Phosphorus in organic matter is inaccessible to plants until decomposers release it. Fungi are particularly important here because they can store phosphorus in their mycelia and release it gradually, preventing it from being washed away by rain before plants can absorb it. But fungi don't only break down dead tissue. Some are parasites, attacking living plants and animals, drawing nutrients from their hosts. These parasitic relationships can be destructive, causing disease and death. But from an ecosystem perspective, parasites play important roles in regulating populations and driving natural selection. A fungus that attacks only weak or stressed trees, for instance, acts as an agent of selection, removing less fit individuals and opening up resources for healthier trees to use. Other fungi form relationships that exist somewhere between mutualism and parasitism. The endophytic fungi, which live inside plant tissues without causing obvious harm, may provide benefits to their hosts under some conditions while extracting a cost under others. The nature of the relationship depends on environmental context, shifting along a continuum from parasitism to mutualism, depending on what resources are available and what stresses the partners face. In agriculture, fungal decomposers are both friend and foe. They're essential for maintaining soil fertility, breaking down crop residues, and returning nutrients to the soil. Composting, the managed decomposition of organic matter, relies heavily on fungal activity to transform kitchen scraps, yard waste, and agricultural residues into rich, dark compost that gardeners prize. But fungi can also attack stored grain, spoil stored fruits and vegetables, and reduce crop yields through disease. The study of fungal decomposition has practical implications that extend far beyond ecology. Understanding how fungi break down lignin has applications in the paper and pulp industry, in biofuel production, in bioremediation of contaminated sites. Some fungi can decompose plastics, petroleum products, and other synthetic materials, that are otherwise extremely persistent in the environment. Researchers are investigating whether these fungi might be harnessed to clean up pollution, to break down landfill waste, to solve the growing problem of plastic contamination in terrestrial and marine environments. The quiet work of decomposition goes on ceaselessly. Every hour of every day, everywhere organic matter exists. Fungi extend their hyphae through dead leaves on the forest floor, through fallen logs in the woods, through manure in a meadow, through the bodies of dead insects, through compost heaps, through countless other substrates. They secrete their enzymes, break down complex molecules, absorb the products, grow and reproduce. Patient, persistent, essential. Without them, life's cycle would be broken, and the world would be a very different place. The largest living thing, in the Blue Mountains of eastern Oregon, within the Malheur National Forest, a few miles east of the small town of Prairie City, something extraordinary exists. It looks unremarkable from above, just ordinary forest, ponderosa pines and Douglas firs growing in the mountain soil. But beneath the surface, spreading through the soil and penetrating the roots of trees, growing in the space between bark and wood, there lives an organism that may be the largest single living thing on Earth. It's a fungus. Specifically, it's Armillaria ostoyi, also known as the honey mushroom or shoestring fungus. And this particular individual, nicknamed the humongous fungus, covers approximately 2,385 acres, roughly 3.5 square miles of forest. To put that in perspective, that's larger than 1,600 football fields. It's so large that it appears on maps of the forest as a single genetic individual, distinct from other colonies of the same species growing nearby. How do we know this is a single organism and not many separate ones? The answer comes from genetic testing. In the late 1980s and early 1990s, when forest pathologists began investigating die-offs of trees in this area, they collected samples of the fungus from many different locations across the forest. When they analyzed the DNA of these samples in the laboratory, using techniques that can distinguish between genetic individuals, they made a startling discovery. The samples from across this vast area were genetically identical, clones of one another, all part of the same mycelial network, all one organism. This discovery overturned the previous record holder, another Armillaria individual from Michigan's Upper Peninsula that covered only about 37 acres. It also forced scientists to reconsider what we mean by concepts like individual and organism when applied to fungi. A mushroom is clearly an individual structure, but what about the mycelium that produces it? If that mycelium extends for miles underground, all connected, all genetically identical, all functioning as a coordinated whole, isn't that also an individual? The weight of the humongous fungus is estimated to be somewhere between 7,500 and 35,000 tons, though these numbers should be taken as rough approximations. Weighing an organism that permeates soil and wood over thousands of acres is not a straightforward task. Even the lower estimate would make this fungus one of the heaviest living organisms on Earth, comparable to the weight of blue whales, though distributed across a vastly larger area. But perhaps even more impressive than its size is its age. By examining the fungus's growth rate and the area it covers, scientists estimate that it could be anywhere from 2,400 to 8,650 years old. The lower estimate would make it contemporary with the building of the Egyptian pyramids. The higher estimate would place its origin in the early Bronze Age, when humans were just beginning to develop writing and build the first cities. This fungus has been growing, slowly and patiently, through all of human history. How does a fungus grow so large? The answer lies in its lifestyle. Armillaria ostoyae is a parasitic fungus that attacks the roots of coniferous trees. It spreads through the forest primarily by growing along root systems, moving from tree to tree through root contact. When its hyphae encounter a suitable host root, they penetrate the bark and colonize the space between the bark and the wood, forming a white, fan-shaped mat of mycelium. As the infection progresses, the fungus girdles the tree, cutting off the flow of water and nutrients, and eventually kills it. But Armillaria doesn't rely solely on moving through roots. It also produces rhizomorphs, specialized structures that look like black shoelaces, hence the name shoestring fungus. These rhizomorphs can grow through the soil for considerable distances, seeking out new hosts. They're tougher and more persistent than ordinary hyphae, able to penetrate obstacles and survive harsh conditions that would kill normal mycelium. When it successfully colonizes a new tree, the fungus can persist there for years or decades, slowly extracting nutrients while the tree gradually declines. The tree may show various symptoms: yellowing needles, dying branches, reduced growth, the oozing of resin from the base of the trunk where the fungus is attacking. But death is often slow, taking decades, and during that time the fungus is growing, spreading its rhizomorphs outward, seeking more hosts. In autumn, when conditions are right, the fungus produces fruiting bodies, the honey mushrooms that give Armillaria ostoyae one of its common names. These mushrooms grow in clusters around the bases of infected trees, golden brown caps on stalks a few inches tall. They're edible, prized by some mushroom hunters, though they must be cooked thoroughly as they can cause gastric upset if eaten raw. The mushrooms produce vast numbers of spores that are dispersed by wind, potentially establishing new colonies far from the parent mycelium. But for the most part, the fungus doesn't rely on spores for local spread. Its expansion is accomplished by the slow, patient growth of rhizomorphs through the soil, the steady colonization of new hosts, the gradual enlargement of its territory at a rate of about 1 to 3 feet per year. At that speed, covering thousands of acres takes millennia. The discovery of the humongous fungus raised practical as well as scientific questions. From a forest management perspective, Armillaria root disease is a serious problem. It kills commercially valuable timber trees. It can create gaps in forests that alter ecosystem dynamics. Forest managers have tried various approaches to control its spread, removing infected trees and stumps, creating barriers by removing roots from the soil, planting less susceptible tree species. But controlling a fungus that's already present across thousands of acres is extraordinarily difficult. Yet from an ecological perspective, Armillaria serves important functions. By killing trees, it creates snags, standing dead trees that provide habitat for countless species of birds, mammals, insects, and other fungi. The gaps created when trees die allow light to reach the forest floor, promoting the growth of understory plants and the establishment of new tree seedlings. The fungus decomposes the dead wood, recycling nutrients back into the ecosystem. In a natural, undisturbed forest, Armillaria is part of the normal cycle of death and renewal. The humongous fungus is not unique. Other very large armillaria individuals have been documented, including one in Washington state that covers about 1,500 acres, and another in Michigan covering 91 acres. These fungi demonstrate that under the right conditions, long-lived clonal organisms can achieve enormous sizes, exceeding any animal and rivaling the largest clonal plant colonies. Indeed, the competition for largest organism is not straightforward. There's a quaking aspen clone in Utah called Pando that covers more than 100 acres and may weigh nearly 13 million pounds, though it's composed of approximately 47,000 separate tree stems, all connected by a shared root system. There are seagrass meadows that extend for miles, all clones of a single original plant. The question of what constitutes the largest organism depends on how you measure and what criteria you use. But perhaps the more important point is this: organisms don't have to be large to be successful. The vast majority of fungi are microscopic, or nearly so. Their mycelia extending through substrates measured in millimeters or centimeters rather than miles. What the humongous fungus represents is not fungal superiority, but fungal possibility. An example of what can happen when an organism finds itself in exactly the right place, with exactly the right resources, with no insurmountable obstacles to its growth for thousands of years. In the Blue Mountains of Oregon, in the soil beneath ponderosa pines, in the space between bark and wood, in the darkness that exists a few inches below the surface of the earth, the humongous fungus continues to grow, slowly, patiently, extending its rhizomorphs outward, colonizing new hosts, recycling the old, maintaining itself through the millennia. A living testament to the secret, hidden grandeur of the fungal kingdom. Nature's pharmacists. On a September morning in 1928, a Scottish biologist named Alexander Fleming returned to his laboratory at St Mary's Hospital in London after a holiday. He had been studying Staphylococcus bacteria, and before leaving, he had left several culture plates on his workbench. When he examined the plates upon his return, he noticed something unusual. One plate had been contaminated by mold, and around the mold, the bacteria had failed to grow. There was a clear zone, a circle of inhibition, where no bacterial colonies existed. Fleming recognized that the mold must be producing some substance that killed or inhibited the bacteria. He identified the mold as belonging to the genus Penicillium, and he named the antibacterial substance it produced penicillin. This accidental discovery, one of the most famous in the history of medicine, inaugurated the antibiotic age and ultimately saved millions of lives. But Fleming's discovery was not the first time humans had used fungi medicinally. For thousands of years, various cultures around the world had employed moldy bread, moldy soy products, and other fungal preparations to treat infected wounds and prevent sepsis. Ancient Egyptians applied moldy bread to wounds. Traditional Chinese medicine included preparations of moldy soybean curd. These practices were based on empirical observation rather than understanding of mechanism, but they worked because the fungi were producing antibiotics. What Fleming did was bring scientific rigor to this folk knowledge. He isolated the mold, cultured it, demonstrated that its broth retained antibacterial activity even after the mold was removed, and showed that the active substance, penicillin, was remarkably effective against a range of bacterial pathogens while being relatively non-toxic to human cells. This selectivity, the ability to kill bacteria without harming human tissue, made penicillin an ideal therapeutic agent. But there was a problem. Producing penicillin in sufficient quantities for medical use proved extraordinarily difficult. The penicillium mold produced only tiny amounts of the antibiotic, and extracting and purifying it was laborious. For more than a decade, penicillin remained a curiosity, known to work but unavailable in quantities sufficient for widespread use. The breakthrough came during World War II, when the urgent need to treat infected wounds in soldiers spurred a massive effort to scale up penicillin production. Researchers in the United States and Britain, working collaboratively, developed methods for growing penicillium in large fermentation tanks and extracting penicillin efficiently. They screened thousands of penicillium strains, looking for ones that produced more antibiotic. The most productive strain was found growing on a moldy cantaloupe in a market in Peoria, Illinois. This strain and its descendants became the workhorses of industrial penicillin production. By 1944, enough penicillin was being produced to treat all Allied forces wounded in the D-Day invasion. The drug proved spectacularly effective against bacterial infections that had previously been fatal: pneumonia, septicemia, gonorrhea, scarlet fever, many forms of wound infection. The impact on mortality was dramatic. Infections that would have killed soldiers now could be cured with a simple course of injections. After the war, penicillin became available for civilian use, and the pharmaceutical industry ramped up production further. The price dropped precipitously as production methods improved and competition increased. A treatment that had cost hundreds of dollars now cost mere pennies. Penicillin and related antibiotics became standard medical tools, used to treat everything from strep throat to meningitis. Fleming, along with Howard Florey and Ernst Boris Chain, who had done the crucial work of purifying and testing penicillin for therapeutic use, received the Nobel Prize in Physiology or Medicine in 1945. Their work had demonstrated that fungi could be sources of powerful medicines, and it inaugurated a golden age of antibiotic discovery. In the decades following penicillin's success, researchers screened thousands of fungi and bacteria for antibiotic production. They found streptomycin, produced by soil bacteria, effective against tuberculosis. They found cephalosporins, produced by fungi, chemically related to penicillin, but with different antibacterial spectra. They found a host of other antibiotics, each produced by some microorganism as part of its chemical warfare against competing microbes. But fungi produce many other medically useful compounds beyond antibiotics. In the 1970s, researchers discovered that a fungus called Tolypocladium inflatum produces a compound called cyclosporine, which suppresses the immune system. This discovery revolutionized organ transplantation. Before cyclosporine, transplanted organs were often rejected by the recipient's immune system, and the drugs available to prevent rejection were toxic and not very effective. Cyclosporine changed that, making successful organ transplantation routine. Fungi also produce statins, drugs that lower cholesterol levels in the blood. The first statin, lovastatin, was isolated from a species of Aspergillus. Statins work by inhibiting an enzyme involved in cholesterol synthesis, and they've become among the most widely prescribed medications in the world, used by millions of people to reduce their risk of heart disease and stroke. Other fungal compounds have found use as antifungal agents, treating infections caused by pathogenic fungi. Griseofulvin, produced by Penicillium, is used to treat ringworm and other fungal skin infections. Echinocandins, derived from fungal products, treat serious systemic fungal infections. The irony of using one fungus to kill another is not lost on scientists, but it makes perfect sense from an evolutionary perspective. Fungi have been engaging in chemical warfare with each other for hundreds of millions of years, and the compounds they've evolved to attack competitors are precisely the compounds we can use as medicines. The mechanisms by which these fungal compounds work are diverse. Penicillin works by interfering with bacterial cell wall synthesis, causing bacterial cells to burst due to osmotic pressure. Cyclosporine works by inhibiting T-cell activation, preventing the immune system from attacking transplanted tissue. Statins inhibit the enzyme HMG-CoA reductase, blocking cholesterol synthesis. Each compound has its own specific target, its own mechanism of action, reflecting the particular evolutionary pressures that led to its production. But why do fungi produce these compounds at all? The answer lies in competition. In nature, fungi must compete with bacteria and other fungi for resources. Producing antibiotics gives a fungus an advantage, allowing it to clear away competitors and claim resources for itself. From the fungus's perspective, these compounds are weapons. From our perspective, they're medicines. The difference is only a matter of application. Modern drug discovery increasingly relies on understanding fungal genetics and biochemistry. The genes that code for antibiotic production are often clustered together in the fungal genome, forming what are called biosynthetic gene clusters. By sequencing fungal genomes, scientists can identify these clusters even in fungi that don't produce antibiotics under laboratory conditions. Then, by expressing the genes in yeast or bacteria, or by manipulating the fungi to activate the genes, researchers can produce novel compounds that might have therapeutic value. This approach has revealed that fungi possess far more chemical diversity than anyone suspected. A single fungal species might have genes for producing dozens of different bioactive compounds, most of which are never expressed under laboratory conditions. The challenge now is figuring out how to access this hidden chemical diversity, how to wake up the sleeping genes and convince fungi to produce compounds they normally keep locked away. The threat of antibiotic resistance makes this work increasingly urgent. Many bacterial pathogens have evolved resistance to the antibiotics we've been using for decades, and some infections have become nearly impossible to treat with existing drugs. We need new antibiotics, and fungi remain one of the most promising sources. The irony is that the very mechanism that makes antibiotics work, their ability to kill bacteria, drives the evolution of resistance. It's an arms race, and we need to keep discovering new weapons. Beyond antibiotics, fungi produce compounds with anti-cancer properties, anti-inflammatory effects, immunomodulatory activities, and a host of other potential therapeutic uses. Some mushrooms, particularly certain species used in traditional Asian medicine, produce polysaccharides that appear to boost immune function. Whether these compounds will prove useful as medicines remains to be established by rigorous clinical trials, but the potential is there. The story of penicillin, from Fleming's chance observation to the massive industrial production that saved countless lives, illustrates the importance of basic research and the potential for unexpected discoveries. Fleming wasn't looking for antibiotics when he found penicillin. He was studying bacteria and happened to notice a contaminant. But because he was observant, because he recognized the significance of what he saw, and because he pursued the observations systematically, he uncovered something transformative. In laboratories around the world, researchers continue to investigate fungi, screening for new compounds, sequencing genomes, manipulating biosynthetic pathways, trying to unlock the chemical diversity that evolution has produced over hundreds of millions of years. Some of this research will lead to new medicines, most won't, but we keep looking because the potential rewards are so great, and because fungi have already given us so much. The molds that grow on bread, on fruit, on damp walls, the mushrooms that fruit in forests and fields, the yeasts that ferment our food and drink. They all carry within their cells the potential for new medicines, new tools, new solutions to the problems that afflict humanity. We need only to look, to ask the right questions, and to pay attention when nature provides an answer. Living light. Deep in the forests of Brazil, on certain nights when the humidity is high and the moon is hidden, hikers might notice something extraordinary. Scattered on the forest floor, glowing softly in the darkness, are small mushrooms emitting a pale green light. The light is faint, barely bright enough to read by if you held the mushroom close to a page. But in the complete darkness of the forest understory, it's startling and beautiful, a natural wonder that seems almost magical. These are bioluminescent fungi, organisms capable of producing their own light through chemical reactions occurring within their tissues. Bioluminescence is common in marine environments, familiar to anyone who's seen fireflies or glowing plankton, but it's relatively rare on land. Among terrestrial organisms, only certain fungi, fireflies, and a handful of other insects and arthropods possess this ability. Scientists have documented approximately 120 species of bioluminescent fungi, distributed across multiple families and found primarily in tropical and subtropical forests, though some occur in temperate regions as well. They belong to several distinct evolutionary lineages within the mushroom-forming fungi, suggesting that bioluminescence evolved independently several times in fungal evolution, or perhaps evolved once early in fungal history, and was retained in some lineages while being lost in others. The light these fungi produce is almost always green, with a wavelength of around 520 to 530 nanometers. This wavelength is in the middle of the visible spectrum, the color to which human eyes are most sensitive under low light conditions. It's also the color that penetrates darkness most effectively, making it visible from the greatest distance. The biochemical mechanism of fungal bioluminescence involves compounds called luciferins and enzymes called luciferases, terms that derive from Lucifer, the light bearer. When luciferin reacts with oxygen in the presence of luciferase, energy is released as light. This is an exergonic reaction, producing light directly without first producing heat, making it extremely efficient. Nearly all the energy goes into producing photons, rather than being wasted as thermal radiation. For many years, scientists didn't know what specific luciferin fungi used, or what pathway produced it. The biochemistry of firefly bioluminescence was well understood, and marine bioluminescence in organisms like jellyfish and certain fish was also well characterized. But fungal bioluminescence remained mysterious. The breakthrough came in recent years when researchers finally identified the fungal luciferin as a modified form of caffeic acid, a compound common in plants and fungi. The luciferin is produced through a biosynthetic pathway involving several enzymes that modify caffeic acid step by step until it becomes the light-emitting compound. When this luciferin is oxidized by the luciferase enzyme in the presence of oxygen and NADPH, it produces light. The reaction is more complex than initially thought, involving multiple steps and intermediate compounds, but the end result is the same: visible light produced by living tissue. But why do fungi glow? What evolutionary advantage does bioluminescence provide? This question has puzzled scientists for more than a century. Various hypotheses have been proposed, but clear evidence has been elusive. The most widely accepted explanation, at least for those fungi whose mushrooms glow, is that the light attracts insects. In the darkness of the night forest, a glowing mushroom is highly visible. Insects, attracted by the light, land on the mushroom. As they walk across its surface, spores stick to their bodies. When the insect flies away, it carries spores with it, dispersing them to new locations where they can germinate and establish new fungal colonies. In this view, fungal bioluminescence serves the same function as the bright colors and attractive scents of flowers, drawing in animals that unwittingly provide pollination, or, in the fungal case, spore dispersal. Support for this hypothesis comes from experiments showing that insects are indeed attracted to bioluminescent mushrooms, and that the insects that visit them carry away spores. In one study, researchers placed glowing mushrooms and non-glowing mushrooms of the same species with luminescence chemically inhibited in the forest and monitored which attracted more insects. The glowing mushrooms received significantly more insect visits, suggesting that the light does function to attract spore dispersers. But not all bioluminescent fungi glow in their mushrooms. In some species, only the mycelium glows, the underground network of hyphae that spreads through decaying wood or soil. What advantage could there be to having glowing mycelium that's hidden from view? Several ideas have been proposed. One is that the glow might be visible through thin layers of bark, or at the exposed edges of decaying logs, still potentially attracting insects. Another is that bioluminescence might be a byproduct of another biochemical process, perhaps related to dealing with toxic oxygen species produced during wood decay, and the light itself might not serve any function. The distribution of bioluminescent fungi provides some clues. They're most common in tropical forests, particularly in regions with high humidity and dense forest canopies that create very dark conditions. In such environments, even a faint light source would be visible and potentially useful for attracting dispersers. The fungi that fruit at night, when darkness is complete, might benefit most from bioluminescence. One of the most famous bioluminescent fungi is Pinellus stypticus, the bitter oyster, which occurs in temperate forests of North America, Europe, and Asia. Not all populations of this species are bioluminescent, however. Populations in eastern North America and parts of Asia glow, while those in western North America and Europe generally don't. This geographic variation suggests that bioluminescence can be lost relatively easily in evolution, perhaps when its benefits don't outweigh its costs. Another well-studied species is Neornithoponus gardneri, found in Brazil. This species produces large, conspicuous mushrooms that glow brightly enough to be visible from several meters away in complete darkness. Local people have reported using the mushrooms as natural torches to light their way through the forest at night. The mushroom's fruit during the rainy season appearing on dead palm fronds and other decaying plant material, and their bioluminescence follows a circadian rhythm, glowing most intensely during the night hours when potential insect dispersers are active. Armillaria species, including the honey mushroom that includes the humongous fungus, are also bioluminescent in some cases. The mycelium and sometimes the mushrooms of Armillaria produce light, and the phenomenon has been observed in forests around the world. Colonized wood glowing with Armillaria mycelium has been called foxfire, a term that derives from folk traditions in various cultures. French folklore called it fairy fire, while sailors referred to glowing wood found at sea as Saint Elmo's fire, though that term more commonly refers to a different phenomenon involving atmospheric electricity. The genes responsible for bioluminescence are clustered together in the fungal genome, forming what researchers call the luz gene cluster, luz being Portuguese for light, since much of the research was done on Brazilian species. This gene cluster contains the genes encoding the enzymes that synthesize luciferin and the luciferase that catalyzes the light-producing reaction. By studying these genes, researchers have been able to trace the evolutionary history of fungal bioluminescence and confirm that it evolved multiple times independently. Interestingly, researchers have successfully transferred the luz gene cluster from bioluminescent fungi into other organisms, including yeast and even plants, causing them to glow. This opens up intriguing possibilities for biotechnology. Imagine plants that light up when they need water, or trees that illuminate pathways without requiring external lighting. Such applications are speculative now, but they illustrate the potential of understanding natural bioluminescence. For the fungi themselves, bioluminescence is energy expensive. Producing luciferins and luciferases requires resources that could be used for growth or reproduction. The fact that bioluminescence has been maintained by evolution in multiple fungal lineages suggests that it provides significant benefits, at least under some conditions. The selective pressure that maintains bioluminescence is likely related to reproductive success, helping fungi disperse their spores more effectively and colonize new habitats. In the darkness of the forest floor, where sunlight never reaches, where the only illumination comes from the faint glow of stars filtered through the canopy above, the bioluminescent fungi shine. Pale green points of light scattered across fallen logs and leaf litter, glowing steadily through the night, a testament to the biochemical ingenuity of evolution. They glow not for our benefit, not for beauty, though they are beautiful, but for their own purposes, recruiting insects, spreading their genes, persisting through time. A fungus doesn't know that it glows any more than a flower knows that it's colourful. These are adaptations, tools shaped by natural selection, but they inspire wonder nonetheless. We who see them in the dark forest might feel that we're glimpsing something normally hidden, a secret of the natural world revealed for a moment before the sun rises and the glow fades away, invisible in daylight but waiting for the next night to shine again. Spore travelers. A single mature mushroom can release billions of spores. A giant puffball can produce trillions. These numbers are almost incomprehensible, but they reflect the fundamental challenge that fungi face. Fungi are immobile, rooted to their substrate by their mycelia. They cannot walk or crawl to new locations. Their only hope for dispersing beyond the immediate vicinity of the parent colony is through their spores, microscopic capsules of genetic material. that can be carried by wind, water, or animals to distant places where, if conditions are right, they can germinate and establish new colonies. Spores are extraordinary structures, marvels of biological engineering. Each spore is typically a single cell, though some species produce multi-celled spores, surrounded by a thick protective wall that shields the genetic material inside from ultraviolet radiation, desiccation, temperature extremes, and other environmental hazards. The wall is often impregnated with pigments, proteins, and lipids that enhance its durability. Some spores can survive for decades in dormancy, waiting for conditions favorable for germination. The diversity of spore structures across the fungal kingdom is immense. Some spores are smooth and round. Others are ornamented with spines, ridges, or reticulations that may help them adhere to surfaces or influence how they move through air or water. Some are coloured, black, brown, yellow, even pink or purple, pigments that protect against radiation or help regulate temperature. Some are tiny, just a few micrometers across, while others are relatively large, visible to the naked eye when clustered together. But producing spores is only half the challenge. The fungus must also disperse them, releasing them into the environment in ways that maximize the chances that at least some will reach suitable habitats. Evolution has produced a stunning array of dispersal mechanisms, each suited to particular ecological niches and environmental conditions. The most common dispersal mechanism is wind. Countless fungal species rely on air currents to carry their spores away from the parent. But getting spores into the air is not as simple as it might seem. There's a physical problem, a boundary layer of still air that exists just above any surface, even in windy conditions. Within this boundary layer, typically just a millimeter or so thick, air movement is minimal, and spores released passively would simply fall to the ground nearby. Fungi have evolved multiple solutions to this problem. Many actively shoot their spores through the boundary layer, forcibly ejecting them with enough velocity to reach the moving air beyond. This is called ballistospory, and it occurs in many groups of fungi, particularly the Basidiomycetes, the group that includes most familiar mushrooms. The mechanism of ballistospory is fascinating. As a spore matures on its supporting structure, a small droplet of fluid called Buller's drop forms at its base. At the same time, a thin film of water accumulates on the spore surface. When these two water masses suddenly merge, the change in surface tension propels the spore sideways with remarkable force. The spore accelerates at more than 10,000 times the force of gravity, reaching velocities of several meters per second, enough to carry it through the boundary layer and into the moving air where it can be transported away. This mechanism operates continuously in a mature mushroom. As you stand beside a fresh mushroom in the forest, millions of spores are being launched from its gills or pores every hour. An invisible artillery barrage that continues day and night as long as the mushroom remains moist and viable. If you disturb a fresh mushroom, you might release a cloud of spores visible as a faint smoke or haze in the air, evidence of the countless spores waiting to be dispersed. Other fungi use different mechanisms to achieve the same goal. Cup fungi and flask fungi contain their spores in structures called aeatis, microscopic sacs that accumulate osmotic pressure as they mature. When an ascus ruptures, it shoots its spores through a small opening with explosive force, launching them into the air. If you pick up a fresh cup fungus and breathe on it gently, providing a bit of moisture, you can sometimes see a small cloud of spores erupt from its surface as thousands of hyphae simultaneously discharge. Puffballs take yet another approach. The mature puffball is a sac filled with dry spores, billions of them, held inside a papery outer skin. The sac has an opening at the top, and when something presses on the sac, a raindrop, an animal's foot, even a gust of wind that causes the puffball to flex, spores are puffed out through the opening like smoke from a bellows. You can trigger this yourself by tapping a mature puffball with your finger, releasing a small cloud of spores that drifts away on the breeze. These mechanical dispersal mechanisms work best in still or moderately moving air. Once spores are ejected into the air above the boundary layer, they become subject to air currents and turbulence that carry them away from the parent. Small spores can remain airborne for hours or even days, traveling hundreds or thousands of kilometers before settling to the ground. Fungal spores have been collected from the upper atmosphere, from high-altitude aircraft, even from the stratosphere, evidence of how widely they can disperse. But wind dispersal is inefficient. The vast majority of spores land in unsuitable locations where they cannot germinate, on rocks, in water, on soil, without organic matter, in environments too hot or too cold or too dry. This is why fungi must produce such enormous numbers of spores. If 1 in a billion spores successfully establishes a new colony, producing trillions of spores gives reasonable odds that some will succeed. Some fungi have evolved dispersal mechanisms that don't rely on wind. Water-dispersed spores have hydrophobic surfaces that make them non-wettable, allowing them to float on water surfaces like tiny boats. These spores are carried by streams, rainwater runoff, or rain splash, traveling shorter distances than wind-dispersed spores, but with more directional movement, following water flows to locations where moisture is abundant. Splash dispersal is particularly common in fungi that grow on dung or on soil surfaces. Rain drops hitting the spore-bearing structures splash the spores into the air, carrying them upward and outward. The bird's nest fungi exemplify this strategy. Their fruiting bodies look like tiny cups or nests containing small egg-like structures called peridioles, which are packets of spores wrapped in protective layers. When a raindrop falls into the cup with the right angle and force, it splashes the peridioles out, sometimes launching them more than a meter away. Animal dispersal is another strategy, particularly important for fungi that produce their spores underground or inside enclosed structures that prevent wind dispersal. Truffles, for example, are subterranean fungi that produce spore-filled fruiting bodies beneath the soil surface. These fruiting bodies emit powerful aromas that attract animals, particularly mammals like pigs, deer, rodents, and bears, which dig up the truffles and eat them. The spores pass through the animal's digestive tract unharmed and are deposited in feces, often far from where the truffle was consumed, providing both dispersal and a nutrient-rich substrate for germination. Insects also disperse fungal spores, sometimes incidentally, and sometimes through more specialized relationships. Some fungi produce fruiting bodies that mimic the appearance and smell of carrion or feces, attracting flies that land on them, pick up spores on their bodies, and carry the spores to new locations. The stinkhorn fungi epitomize this strategy, producing mushrooms that smell like rotting meat and secrete a slimy, spore-laden substance that sticks to visiting insects. Other fungi form mutualisms with specific insects. Some bark beetles carry fungal spores in specialized pouches on their bodies, inoculating the wood they tunnel through with the fungus, which then decomposes the wood and provides food for the beetle larvae. Some ants and termites actively cultivate fungi, maintaining fungal gardens that they fertilize and tend, harvesting fungal tissue for food. These relationships are ancient and highly co-evolved, with some ant species completely dependent on their fungal partners for nutrition. The duration that spores remain viable varies enormously. Some spores are short-lived, able to germinate only if they reach a suitable substrate within days or weeks of release. Others can remain dormant for years, even decades, waiting for the right combination of moisture, temperature, and nutrients to trigger germination. Spores found in ancient peat bogs, preserved in the anoxic environment, have been germinated in laboratories, demonstrating viability after thousands of years. When a spore finally lands in a suitable location and conditions are favorable, it germinates, sending out a tiny hyphae that begins to grow and branch. This initial growth is precarious. The young mycelium has only the stored nutrients within the spore to sustain it until it can establish connections with a food source. If it cannot find nutrients quickly, it will die. But if it succeeds, if it finds sufficient food and the environment remains hospitable, it grows, spreading its hyphae, establishing its network, and eventually, when mature and when conditions align, producing its own fruiting bodies and releasing its own spores, continuing the cycle. In every breath you take outdoors, you inhale fungal spores. They're everywhere, invisible, abundant beyond measure. Most will do nothing, settling on surfaces where they cannot grow or being destroyed by immune cells if they enter your lungs. But some will find purchase, will germinate, will begin the slow process of building a mycelium and contributing to the endless work of decomposition and nutrient cycling that makes terrestrial ecosystems function. The air is full of potential, full of genetic material, traveling from place to place, seeking the circumstances that will allow it to express itself, to grow, to reproduce, to continue the lineage. This is the realm of the spore travelers, the dispersing stage of the fungal kingdom, invisible but essential, linking generations, connecting habitats, maintaining the diversity and ubiquity of fungi across the globe. Extremophiles of the fungal world. When we think of extreme environments, we typically picture places hostile to life. The frozen wastes of Antarctica, the superheated water of volcanic hot springs, the hypersaline pools of salt lakes, the desiccated rocks of deserts, the acidic runoff from mines, the depths of the ocean where pressure is crushing and light never reaches. These are places where life, we assume, must struggle to exist, if it exists at all. But life is extraordinarily tenacious, and fungi are among the most versatile and resilient organisms on the planet, capable of colonizing virtually every environment Earth offers. Consider Antarctica, the coldest place on Earth, with temperatures that can drop below -90 degrees Celsius, with months of darkness, with fierce winds and virtually no liquid water. This seems like a place where nothing could live. Yet fungi thrive there, growing inside rocks in the dry valleys, one of the most Mars-like environments on Earth, persisting as members of crypto-endolithic communities, life forms that live within the porous spaces inside rocks, where they're protected from the worst of the environmental extremes. These rock-dwelling fungi form black or dark green patches just beneath the rock surface. They're visible when you crack open a rock, revealing the colored layers within. The fungi are often associated with photosynthetic algae, forming lichen-like communities that can photosynthesize during the brief periods when conditions allow and survive in dormancy the rest of the time. Some of these fungi can tolerate being frozen and thawed repeatedly, can survive extreme desiccation, and can resume growth after being dormant for months or years. How do they survive? Part of the answer lies in specialized adaptations at the cellular and biochemical level. These fungi produce compatible solutes, small organic molecules like glycerol, trehalose, and polyols that protect cellular structures from damage during freezing and desiccation. These compounds act like cellular antifreeze, lowering the freezing point of cell contents and preventing the formation of ice crystals that would rupture cell membranes. They also stabilize proteins and membranes when water is scarce, preventing them from denaturing or breaking apart. The fungi also produce melanin and other pigments that protect against ultraviolet radiation. In Antarctica, UV levels are extremely high, especially during the austral summer when the ozone hole is present. Without protection, DNA and proteins would be destroyed by UV damage. The melanin absorbs UV light, dissipating its energy harmlessly, shielding the vital cellular machinery beneath. But cold environments are just one category of extreme. At the opposite end of the temperature spectrum, some fungi live in hot springs where water temperatures exceed 60 degrees Celsius. These thermophilic fungi have enzymes and cellular structures that remain stable and functional at temperatures that would denature the proteins of most organisms. Their cell membranes have different lipid compositions that remain fluid at high temperatures rather than becoming too rigid or breaking down. One of the most studied groups of thermophilic fungi includes species found in composting organic matter, where microbial activity generates considerable heat. These fungi can grow at temperatures of 50 degrees Celsius or higher, thriving in the hot, steaming heart of a compost pile, where most organisms cannot survive. They play crucial roles in breaking down plant material in these environments, working alongside thermophilic bacteria to decompose organic matter rapidly. Salinity presents another extreme challenge. Most organisms cannot tolerate high salt concentrations because salt draws water out of cells by osmosis, causing dehydration and cellular dysfunction. Yet halophilic and halotolerant fungi can survive and even thrive in environments with salt concentrations far exceeding that of seawater. These fungi are found in salt marshes, salt lakes, salterns, and even on salted food products. Research has revealed that halophilic fungi employ multiple strategies to cope with salt stress. They can accumulate compatible solutes internally to balance the osmotic pressure from external salt, preventing water loss. They can modify their cell walls to reduce water loss. They can actively pump excess salt out of their cells using specialized membrane proteins. Some can even alter their metabolism to function efficiently under high salt conditions. One well-studied halophilic fungus is Hortea wernecki, a black yeast that grows in salt marshes and salterns around the world. This fungus can tolerate salt concentrations up to 5 times that of seawater, growing happily in environments that would kill most organisms within minutes. Studies of its genome and biochemistry have revealed sophisticated regulatory systems that sense salt levels and adjust cellular physiology accordingly. Acidity and alkalinity also pose challenges. Most organisms function best at neutral pH, around 7, but some environments are extremely acidic, with pH values below 3, like acid mine drainage, where sulfuric acid leaches from exposed rocks. Other environments are highly alkaline, with pH values above 10, like soda lakes, rich in sodium carbonate. Both extremes disrupt cellular processes, denaturing proteins and damaging membranes. Yet fungi can be found in both acidic and alkaline environments. Acidophilic fungi grow in acid mine drainage, on acidic soils, in the acidic digestive systems of some animals. Alkalophilic fungi grow in soda lakes, in alkaline soils, in sewage treatment systems where pH is elevated. These fungi have evolved mechanisms to maintain internal pH near neutral, despite external extremes, using membrane pumps to move protons in or out of cells as needed, buffering their cytoplasm against pH changes. Radiation resistance is another remarkable fungal capability. At Chernobyl, in the ruins of the nuclear reactor that melted down in 1986, fungi are thriving. Black pigmented fungi have been found growing on the walls of the reactor itself, in areas where radiation levels are lethal to humans. These fungi not only tolerate the radiation, but appear to use it, employing melanin to capture energy from gamma rays in a process analogous to photosynthesis, though far less efficient. The mechanisms of radiation resistance are not fully understood, but they involve rapid DNA repair systems that can fix radiation damage, protective pigments that absorb radiation, and cellular structures that are simply more resistant to damage. Some of these fungi grow toward radiation sources, suggesting they're actively seeking out the energy, though the phenomenon remains controversial and requires further study. Pressure is another environmental extreme. In the deep ocean, at depths of thousands of meters, pressure can exceed 1,000 atmospheres, enough to crush most familiar organisms. Yet fungi have been isolated from deep-sea sediments, from the guts of deep-sea animals, even from hydrothermal vents, where hot fluids rich in chemicals erupt from the seafloor. These barophilic fungi have cellular structures adapted to function under extreme pressure, with membranes and proteins that remain stable and functional in conditions where surface-dwelling organisms would be destroyed. Desiccation tolerance is perhaps one of the most widespread extreme adaptations among fungi. Many environments experience periods of severe drought when free water is unavailable for days, weeks, or months. Fungi in these environments can enter dormancy, their metabolism slowing to nearly zero, their cells protected by compatible solutes and protective proteins. When water returns, they rehydrate and resume growth as if nothing happened. Some fungi can survive being completely dried out, their cells containing less than 10% water by mass for extended periods. Spores are particularly resistant, designed specifically to withstand desiccation during dispersal. But even vegetative mycelium can sometimes tolerate extreme dehydration, forming resistant structures called sclerosia, hard knots of compacted hyphae that can survive for years in dormancy. The ability to survive extremes often comes at a cost. Organisms adapted to extreme environments frequently grow more slowly than their counterparts in benign conditions, because they must divert resources into protective mechanisms. They may have more limited metabolic capabilities, unable to utilize as many different food sources. They may be less competitive when conditions are moderate, outcompeted by organisms that don't bear the metabolic burden of extreme adaptations. But in the places where they live, these extremophilic fungi are champions. They colonize niches that others cannot, exploiting resources that others cannot access, surviving conditions that would kill others. And in doing so, they expand our understanding of what life can be, of how far the boundaries of habitability extend. The study of extremophilic fungi has practical applications beyond pure science. These organisms produce enzymes that function under extreme conditions, enzymes that have industrial value. Heat-stable enzymes from thermophilic fungi are used in industrial processes that require high temperatures. Salt-tolerant enzymes could be used in processes involving high salt concentrations. Understanding how extremophiles protect themselves from radiation or desiccation could inform strategies for protecting human cells from similar stresses. Extremophilic fungi also serve as models for astrobiology, the study of potential life beyond Earth. If fungi can survive in Antarctic rocks, in salt lakes, in radioactive ruins, perhaps life could survive in the harsh environments found on Mars, on the moons of Jupiter and Saturn, on other worlds in the universe. By understanding how life adapts to extremes on Earth, we can better predict where we might find it elsewhere and what forms it might take. In the coldest, hottest, saltiest, most acidic, most alkaline, most radioactive, most pressurized, most desiccated environments on Earth, fungi persist. They grow slowly, reproduce when they can, survive when they must, adapting and enduring. They remind us that life is not fragile, not confined to the narrow band of comfortable conditions that humans prefer, but remarkably robust, extraordinarily versatile, capable of flourishing in places that seem impossible. The fungal kingdom spans not just the temperate forests and fertile soils we consider normal, but extends into the extremes, the edges, the places where survival itself is an achievement. The night has grown deep around us now. We've traveled through vast underground networks, witnessed the slow growth of ancient organisms, discovered medicines in mold, watched light emerge from living tissue, followed spores on their journeys, and visited the harshest environments Earth offers. Throughout all of it, fungi have been our guides, revealing a kingdom that exists mostly hidden from view, but that underpins the very possibility of life on land. When you walk through a forest tomorrow, remember the mycelium beneath your feet, connecting the trees, cycling nutrients, supporting life. When you take an antibiotic, remember the fungi that produce these medicines, engaging in chemical warfare that we have repurposed to save human lives. When darkness falls, remember that somewhere in some forest, fungi are glowing softly, calling to insects in the ancient language of light. And remember that in the harshest places, where no other life can persist, fungi endure, testament to the resilience and creativity of evolution. The hidden kingdom of fungi is all around us, invisible but essential, ancient but constantly renewing, diverse beyond our complete understanding, and absolutely fundamental to the world we inhabit. We've barely begun to catalogue its species, to understand its biochemistry, to appreciate its ecological roles. Every year brings new discoveries, new species, new compounds, new relationships, new wonders. The fungi are patient teachers, and they have much yet to teach us, if we're willing to look, to listen, to pay attention to the quiet work happening in the soil, in the wood, in the darkness, all around us. If you enjoyed this journey into the hidden kingdom, please like this video and subscribe to Bedtime Biology. Share your thoughts in the comments below. Tell us where you're listening from, what aspect of fungi fascinates you most, or what other topics you'd like us to explore in future episodes. Until next time, sleep well, and remember, the world is far more interesting, far more interconnected, far more alive than it appears at first glance. Good night.